All right, let's jump right in. Inside every single bacterium, there's this incredible microscopic library of instructions. It's DNA. And today, we're going to figure out the really elegant way it finds the exact right page in that library and, well, fires the starting gun for reading a gene.
I mean, think about it for a second. A bacterium like E. coli has thousands and thousands of genes, but it definitely doesn't need them all turned on at once. So it needs a super precise system to say, okay, read this gene right here, right now. How on earth does it get that specific?
So the main character in our story today is this amazing molecular machine called RNA polymerase. Its whole job is to move along the DNA and copy a gene's instructions into a message, which we call RNA. But as we're about to see, it has a pretty critical weakness.
You see, this polymerase is actually a two-part system. On its own, we call it the core enzyme. And it's a really powerful engine for sure. It can make RNA like crazy, but it's completely blind. It has no idea where the starting points for genes are. Now, when it gets its guide, it becomes the holoenzyme, the complete precision machine. And some early experiments really made this crystal clear. Just take a look at the data here. In that top row, when scientists gave the core enzyme a specific perfect gene to read, its activity was, well, it was practically zero. Look at that, 0.5. It was totally lost. But the holoenzyme, its activity is a whopping 33.0. It knew exactly where to go. So what's that crucial missing piece?
Well, it's this, the sigma factor. The easiest way to think about it is like a gene positioning system, a GPS for the cell. It's this small protein that latches onto the blind core enzyme and tells it exactly where to start working.
And this quote just sums it up perfectly. Without sigma, the core can still make RNA, yeah, but it just does it randomly all over the place. It totally lacks specificity. So sigma is the absolute key that unlocks targeted gene expression. Okay, so if sigma is our GPS, what coordinates is it actually searching for on that huge map of DNA? What does the starting line of a gene even look like to a molecule? It's looking for a very specific DNA sequence called a promoter. This is the official docking station, the designated starting line, right before a gene that basically flashes a giant sign saying, hey, polymerase, start reading right here.
And a promoter's address has two key parts, two specific sequences of DNA's chemical letters. So about 35 letters or bases before the gene actually starts, there's a sequence we call the -35 box. And then about 10 bases before, there's the -10 box. The sigma factor is literally built to recognize and grab onto both of these spots.
The whole docking process happens in three main stages. First, the complete holoenzyme just kind of scans along the DNA. When it finds a promoter, it binds, forming what we call a closed complex, because the DNA is still zipped up. Then, this is where the magic happens. The polymerase pries open about 17 base pairs of that DNA, creating an open complex. The track is now officially open for reading. And would you look at the difference sigma makes in just binding strength alone? Some classic research showed that the core enzyme by itself falls off the DNA in less than a minute. But the complete holoenzyme, it latches onto that promoter so tightly, it can stay there for 30 to 60 times hours. This super tight grip is absolutely essential for what comes next.
But this creates a fascinating new problem, right? A paradox. The polymerase has to bind incredibly tightly to find the right spot and to pry the DNA open. But to actually read the gene, it has to move. So how does it break those powerful bonds and escape the starting line it just worked so hard to find?
Well, at first it doesn't. The machine actually gets stuck, kind of like a car engine sputtering. It starts making these tiny little RNA fragments, just a few letters long, but then it fails to move forward and just spits them out. This is called abortive initiation. The polymerase is revving its engine like crazy, but it just can't get out of the starting block. The solution is this incredible bit of molecular gymnastics called DNA scrunching. It's so cool. Instead of the polymerase itself moving forward, it stays put and pulls the DNA from ahead into itself, kind of like you'd pull a rope towards you, hand over hand. So here's how this great escape works. The polymerase holds its position, locked onto the promoter. Then it starts scrunching, just pulling that DNA template into its active site. This scrunching action builds up a massive amount of tension and stores energy, just like when you compress a spring. And finally, all that stored energy is released at once. It's so powerful it breaks the polymerase's bonds to the promoter and literally launches it down the DNA track. What's just amazing about this is that the energy that it needs to escape doesn't come from some external fuel source. It comes from the failed attempts themselves. The scrunching process harnesses the energy from all those little sputtering starts to power the one big successful launch. It's just incredibly efficient.
And with that, it's off. The core enzyme is now free of the promoter, and it can finally do what it does best: race down the gene, reading the DNA code, and building that RNA message.
But hang on a second. What about our guide, the indispensable sigma factor? Its whole job was to get the polymerase to the starting line. Now that the race has begun, is it still needed? Well, the cell has this beautifully efficient system for this. The sigma factor actually gets recycled in a process we call the sigma cycle.
As soon as the polymerase has cleared the promoter and is on its way, the sigma factor just pops off. It's now free to go find another blind core enzyme that's just floating around and guide it to another gene's starting line. This cycle allows a single sigma factor to kick off transcription over and over and over again. It's molecular multitasking at its absolute finest. And as that core enzyme is speeding along, it maintains this tiny moving little bubble. If you could zoom in at any given moment, you'd see this temporary little hybrid of about 8 or 9 base pairs where the new RNA is still attached to the DNA, right before it peels off. It's a fleeting but totally essential connection.
So let's just recap the whole journey. The cell solves this huge problem of finding one gene among thousands by using a two-part machine, a blind but powerful core enzyme, and its precision GPS, the sigma factor. They bind super tightly to a promoter, use the energy from DNA scrunching to escape, and then they recycle the sigma factor to do it all over again. What a process. This unbelievably elegant process is happening countless times every second inside living cells all around us and inside us right now. It's just one of who knows how many molecular machines that make life possible. It really makes you wonder, doesn't it? What other amazing secrets are just waiting to be discovered inside the tiny machinery of the cell?